And this room here is our GC laboratory that's dedicated to the GC of our samples. We have 12 of these in this room, and each one of these instruments has an auto sampler to handle any and all of the samples that we process on a daily basis. In this room, we have part of our QC department. Dedicated in this particular room is all of our reverse phase equipment used for purity analysis. We have 85 Agilent pieces of equipment dedicated towards the analysis of the purity of all of our products. We also have UPLC products available for analysis of peptides and oligonucleotides in our R&D. In addition to our facilities for HPLC purity, we also have in this particular facility 4 of our LC-MS pieces of equipment that are dedicated to structure elucidation and or sequencing of peptides or oligonucleotides. This is a real beneficial piece of equipment to utilize for the quality control and verification of the proper structure or sequence of the products that we've made, and it's particularly advantageous to our GMP client. Additionally, we have in another room GCMS, which is also useful for the same purpose but for more volatile compounds. The final part of the lyophilization process of following or purification is we take the product, we freeze it, and we place it on these vacuum manifold lyophilizers. Here you see a few products that we're lyophilizing, even a nice yellow colored one, which is indicative of a fluorescent dye that's positioned on the peptide for people to use it as a probe. Also, in the back of this room, you see our larger scale shelf lyophilizer for larger quantities so that we can do individual binding as well. This is our purification lab, which represents the penultimate step of the process of peptide manufacturing. Ultimately, the peptide is run on one of our larger columns here in our R&D lab, which is a 20-centimeter column in an axial compression setup so that you can achieve maximal pressure on the system and increase the overall purification theoretical plates, allowing us to purify the peptide from closely adhering impurities to reach the target purity level desired by the customer and whatever their demands may be. When we finish purification on our large-scale 20-centimeter columns, we take that and we analyze the fractions that we've collected, and we do that on what we call our in-process HPLC equipment that you see here. And this is an example of what we're trying to achieve, which is a high level of purity where the impurities have been removed and are nearly at a baseline level, and a highly pure product is seen by a single symmetrical peak emitting from the HPLC column. When we purify oligonucleotides, we use a slightly different property where the oligonucleotide has multiple negative charges. So we use ion exchange as a way to purify the product and remove the closely impurities that have formed from synthesis. These are primarily the N-1 and N+1. You can see such a purification shown over here on the screen where the product is eluted, and the front impurities are typically the N-1 and the back impurities are the N+1, and we collect fractions up here using this system such that we can isolate the desired product with a suitable quality. In this lab, you can see something that makes Jiuzhou Pharma truly unique. We are capable of producing cytotoxic drug conjugates. In this particular room, you can see the multiple isolation chambers and glove boxes used for working with the R&D levels. The final step in the isolation of our cytotoxic drug conjugates requires lyophilization. And you can see such our setup here for R&D cytotoxic substances with our isolation chamber and lyophilizer that's specific for these types of products. Step in the isolation of our cytotoxic drug substances and drug products is handled by an isolated lyophilizer that you see here in this isolation chamber. This particular lyophilizer handles our R&D samples, and we have a scale-up version of this down in our Jiuzhou facility. In this room, you see one of our several instruments of our 400 megahertz NMR. We use this for structure identification and confirmation. You can see that this particular instrument has the capability of handling multiple samples, each individually. It can be utilized to run high-performance quality spectra. The system is maintained under liquid helium, which you use to cool the magnet, and this particular piece of equipment is utilized in our QC department for structure verification. Here you see in this room our standard way of salt exchanging our peptide products. We use a reverse phase process where you can see that this column is isolated from our initial upstream purification part of the process. Prior to that, we take the product into here, we load it on a reverse phase column, we do a base wash step, and after the base wash step, we exchange that into the appropriate buffer. It can be an acetate, it can be a hydrochloride salt, whichever the customer may desire. Even sodium salts are relevant to some of these new GLP agonists. After we've finished the purification and salt exchange, the final step is in the back corner here. You can see that we have a filtration device, so we can filter the final product through a 0.2 micron filter in order to eliminate any bacterial spores or endotoxins.
Behind me, there are 2 rooms. So these 2 rooms are dedicated for the lyophilizer process. For this room is for the engine part of the lyophilizer. And this area is Zone C area. We have the door opened in the Zone C area. After the drying process is finished, we take out the product from the door and put it on the dry glove box to perform the packaging. Because the product is humidity sensitive, so when we perform the packaging, we will have the nitrogen to protect the product and to reduce the humidity.
Okay, so the area behind me is the place where we keep our raw materials and finished products. So depending on the nature of the product, so we could keep them under different temperatures, including 2 to 30 degrees, room temperature, 2 to 8 degrees, and minus 20 degrees. So we kept them in different separate areas. Okay, I'm standing by the example of our cold storage. So the temperature for this storage is 2 to 8 Celsius degrees. The location I am standing right now is a room where we keep the temperature 2 to 30 Celsius degrees.
And here you see our Jiuzhou site, the tank farm that's located here. This particular tank farm feeds all of our different production facilities, but the peptide unit would use primarily our DMF, methanol, and isopropanol. You see the individual piping that takes each of the solvents to the different production facilities. And with this, we have a fully automated system for delivering solvent as needed to each one of our production areas.